Bismillahirrahmanirrahim. Welcome back, friends. Welcome to another video lecture by Kami Biology. Today in this video lecture we are going to talk about pathogen recognition system. Okay, in the previous video lecture, if you guys remember, we talked the phagocytosis phenomena, the overview of the phagocytosis. Okay, this is another video lecture which is actually regarded with the phagocytosis, and in this video lecture what we are going to talk about, pathogen recognition system. So let me write. Now what actually happened in this process? In this process, the bacterial cells are detected, or we can say the bacterial cells are engulfed by the phagocytes. Okay, the phagocytes are not engulfed our own cells. They are usually engulfed the bacterial cells. They are usually engulfed the microorganisms. Now what the reason behind that? Why they are not engulf our own cell and why they are engulfed the microorganism? The reason behind that, on the surface of the bacterial cell there are some special molecules, some special proteins, some special lipids, which we usually call it phospholipids, glycoproteins, we call it Piptidoglycan, Lipopolysaccharides, xamosines, doubles, different kinds of molecules are present on the bacterial or on the microorganism surface. On the other hand, our own cells means on the other hand, on the surface of the phagocytes, the phagocyte contain different kinds of receptors, which we usually call the MHC2 receptor, MHC1 receptor, toll-like receptor, Mannose receptor, Lipopolysaccharide receptors. So all these receptors, they are present. Due to the presence of these receptor and due to the surface molecule which is present on the bacterial cell, an interaction is usually happen. And due to such interaction, we usually call it pathogen recognition. Okay, so due to this way, the phagocytes are usually interacted with the bacterial cell. On the host cell, such molecules are not present, as I explained in the microorganism. So such molecules are not present on our host cell. That's why our host cells are not detected, but the bacteria or any microorganisms may be detected by the phagocytes. Whenever, remember one thing, whenever the phagocytes are attacked on the host cell, then it is actually autoimmune disease. It will be pathological condition. But actually here I am explaining the physiological phenomena. So let me explain. First of all, let me draw the cell structure. That let's say this is a macrophage. So let's say this is a macrophage. So on the surface of the macrophage, let me draw different kinds of, this is maybe a macrophage, phagocytes, any phagocytes. So let's say these are the phagocytes. On the surface of the phagocyte there are different kinds of receptors. So let me draw each of the receptors. Now this is the first receptor, which we usually call that Mannose receptors. They contain another receptors which we usually call that now this is another receptors. Okay, this receptor are called as LPS receptor, Lipopolysaccharide receptor. They also contain usually called that MHC1 receptors. They are also contain MHC2 receptors. Usually MHC2 receptor are not present. MHC2 receptor may be formed when the microorganism are converted into small fragment. After that, MHC2 receptor are formed and they will express each fragment. Okay, but sometime in some books they says that MHC2 receptor are also contained. So MHC2 receptor are also contained, but usually it is not present. Okay, it may be formed when the bacteria are converted into small fragment. So let's say they contain MHC2 receptor as well. So let's say this is MHC2 receptors. They also contain lipid receptor. Moreover, they are also contain this is another receptor which we usually call it lectine receptor. They also contain glycoprotein receptors. Moreover, they are also contain RGD receptors. So let's say this is the RGD receptors. They are also contain toad-like receptors. So here I am not drawing the toad-like receptors because there are 10 classes of the toad-like receptor. Inshallah, I will explain the toad-like receptor in the next video lecture. Okay. Now, due to the presence of these receptors on the phagocytes, we call that PRR receptors. What? All these receptors. PRR. Pattern recognition receptor or pathogen recognition receptors. Okay, these are all pathogen recognition receptors. Why? Because they can recognize the pathogens. Now, there are different kinds of interactions happen between the bacterial cell and our phagocytes, and there are different name of each interaction. First of all, we have an interaction usually formed, which we call that lectine carbohydrate interaction. So let me write lectine carbohydrate interactions. Okay, in this first interaction, the lectine of the bacterial cells, means the fimbrea of the bacterial cell, and the glycoprotein of the amino cells are interacted with each other. Okay, so if let's suppose this is the glycoprotein, they usually detect the lectines. Okay, so these are, let's suppose these are the lectine, or these are the fimbrea of the bacterial. Now this is, let's suppose the bacterial cells, and these are the fimbrea. Okay, if you don't know about the fimbrea, so I already explained it. Fimbrea are actually thin, small hair-like structures, okay, which are specially used for the attachment. Now these fimbrea are detected by this glycoprotein. Okay, lectine carbohydrate interaction. In this interaction, they are also include the interaction between lectine and polysaccharide of the bacterial cells. Okay, now this lectines usually detect what? This lectine usually detect the polysaccharide. If let's suppose this is again a bacterial cells, now at that outer area, as you are watching that outer area, these are called capsule, or these are the polysaccharides layer. Okay, I already explained the capsule as well. You can watch it into my channel. Now this capsule is detected by this lectines. Okay, so the detection of this fimbrea and this polysaccharide by this lectine and this glycoprotein, each interactions are called as lectine carbohydrate interaction. This is the first interaction. There is another interaction which are also happen in the bacterial cell. We usually call that protein-protein interaction. So let me write. These interactions are happen between the RGD, means the arginine glycine molecules, arginine glycine aspartic acid of the bacteria, and this interaction are also happen between the receptor of RGD as well. So let's suppose this is the RGD receptor of the phagocytes. So this RGD receptors usually recognize the aspartic acid, means arginine glycine aspartic acid, or we also call it RGD. Okay, so let's suppose again, if this is the bacterial cells, so at that specific area, outer surface area of the bacterial cell, they contain arginine glycine aspartic acid. Let me write in a short form: arginine glycine aspartic acid. Okay, so this molecules are detected by this RGD receptor, and the interaction between them is called as protein-protein interactions. Okay, third, we have another interaction which we usually called as hydrophobic protein interaction. Now this interaction are usually happen between the glycolipids of the bacteria, means the lipopolysaccharide acid of the bacterial cell, and the lipid receptor of our amino cells, means our phagocyte cells. So if again this is a bacterial cells, let me draw again the bacterial cell here. If again this is let's suppose a bacterial cells, at the outer surface, if you remember, I already explained this, teichoic acid On the outer surface of the bacterial cell, there are a large fatty acid chain, which we usually call that teichoic acid, lipoteichoic acid. This lipoteichoic acid are usually detected by these lipid receptors. Okay, so they will detect these kinds of molecules, and we call this interaction hydrophobic interaction. Now, all these molecules which are present on the bacterial surface and which are detected by the phagocytes, we call these molecules PAMPS. What we call these molecules? We call each molecule PAMPS. Now, what PAMPS stand for? Pathogen associated molecular patterns. Okay, each molecules are present on the bacterial surface, and the molecules which are involved in this case, these are the flagella, these are the RGD, means arganin, glycine, aspertic acid, these are the Lipopolysaccharide, these are the lectine, fimbrea, different kinds of the surface molecules, and we call that PAMPS. Okay, moreover, this Lipopolysaccharide receptor, as you are watching, they will usually detect the Lipopolysaccharide which are present in case of the gram-negative bacteria. So let's suppose if this is the gram-negative bacteria. So this receptor usually detect the gram-negative bacteria, because on the surface of the gram-negative bacteria there are the Lipopolysaccharide, and this one means this glycoprotein usually detect the Piptidoglycan layer of the gram-positive bacteria. Due to this interaction, they will activate innate immune system. So they usually activate innate immune systems. Okay. Now, one thing more about this Mannose. This Mannose receptor usually detect the Mannose which are present on the fungi cells. Okay, so on the fungi cell wall there are the Mannose. So this receptor usually detect the Mannose. Okay, or okay, I think everything is done. Yes. If still there is anything missing, so then you can write it into a comment. Inshallah, I will explain it. Okay, so that's all about the pathogen recognition systems. Okay, I hope you understand about this video lecture. Inshallah, in the next video lecture, we are going to talk about the toll-like receptor, which we not mention here because it is a separate and a complete lectures, and it contains 10 classes toll-like. So we have 10 classes of the toll-like receptor. That's why I not mention here. So inshallah, in the next video lecture, we are going to talk about toll-like receptor. So please stay tuned and continue to watch the video. Thank you so much.